Hello everyone, and welcome to a new episode and a new series. This new series will explore the physiology of animals, organisms within the kingdom Animalia. Today's show is episode 67, and I'll be exploring animal development, starting with the fertilization of a new individual animal and going through its developmental processes until it reaches sexual maturity and can reproduce on its own, to perpetuate its lineage and its genes forward through time. The kingdom Animalia is awe-inspiring in its extreme diversity and uniqueness, even though basically all animals are built around the same fundamental premise. Now, I'll get into the physiological details more later in the episode, but for now, I'm going to describe them in the most general mechanistic terms possible. And I want to do this so that you can kind of get a sense of how animals are described and understood physiologically, anatomically, biologically. And in that sense, animals are mobile feeding platforms that take a general tube shape. The animal will acquire food and put it in its mouth, where the food will then pass through the tube, which is the digestive tract. The animal will extract nutrients from the food, and then it will excrete the waste out of a second orifice at the end of the tube. Some animals don't have a second orifice, and they just excrete their waste through the same opening that they consumed the food through. This is the case with, for example, jellyfish. Now, all animals, with their basic tube-like body shape, have evolved all manner of limbs and protrusions and sensory organs to detect the world around them, and to move within it, and manipulate and engage with the world around them. Their adaptations help them avoid predators, other animals that would attack their bodies and destroy them and kill them, so that animal could eat them, eat their flesh for chemical energy. They have adaptations that help them capture food to put in their own tube, which will sustain their body, at least until they need more food. And if they can successfully eat food regularly and stay alive, if they can maintain their physical body, their corporeal form, at least until they mature, and they might get a chance to reproduce and create offspring of their own. Now, this is where the story of animal development really begins. Because virtually all animals are sexual organisms, it takes the mating of a male and female parent to produce a fertilized egg, or a zygote. When two sexually mature individuals meet out in the wild, they might engage in some kind of mating ritual or show-off behavior, and if the initiating party is successful in their mating behavior, and the receptive party approves that they're like, yeah, okay, I will mate with you, then they will have sex. And during this act, the male will deposit his gametes in the form of sperm into the female. The individual sperm will compete to navigate the female's reproductive tract, following chemical cues to find and fertilize an egg. When the sperm meets the egg, the sperm first has to burrow through an outer jelly-like layer before it actually reaches the egg's membrane. Now at that point, a chemical process occurs, where the sperm will bind to the egg's outer membrane. And this will also chemically change the rest of the egg's membrane. The whole thing will change so that no further sperm can bind to it. The sperm is then absorbed into the egg's membrane, where it deposits the father's genetic contribution, this haploid genetic material, into the egg's cytoplasm. The sperm's haploid genome will be attracted to the nucleus, where it will find and fuse with the egg's haploid genome. And this will create a fertilized diploid egg. This is the beginning of a new individual. An important thing to note here is that both the sperm and egg of any given species are coated with proteins that act as species-specific markers. The sperm cells express proteins on the outer surface of their membrane that act as ligands. This is to say that they bind to stuff. And the egg cells express a complementary suite of protein receptors that they express on their membrane. These receptors bind to the ligands that the sperm expresses, and when that binding takes place, the egg can recognize that it's bound to a species-appropriate sperm, and it can then initiate plasmogamy, or the fusion of the membranes, to allow the father's genetic contribution to move into the cytoplasm. What's important here is that these species-specific binders and receptors, expressed on the sperms and eggs, respectively, prevent crossbreeding with organisms of another species. If it's another species, especially a distantly related one, then the types and the amount and the pattern of these membrane proteins on the sperm and egg will be different. They'll be so different that they'll be completely incompatible. and the egg can't recognize the sperm, so it won't bind, it won't undergo plasmogamy, and the male's haploid genome will not enter the egg. Now, this barrier is known as a prezygotic block to reproduction, and it prevents the birth of hybrids, and thus it encourages speciation. Now, I talked about this in a lot more detail in the Intro to Evolution series, so if you want to go check that out, I believe that's Series 4. Anyway, when fertilization occurs, the process of development really begins. The fertilized egg will divide, once, and then twice, and then a third time, then again, and it'll produce a ball of cells. Now, this process has some of the fastest rates of cell replication in the organism's entire life. The cells here will replicate on a time span of minutes, and they can produce many tens of thousands of cells in a single day's time. All of the increasingly small cells produced by this rapid division are called blastomeres, and they're part of a structure called a blastula. This blastula is the very beginning of the new individual, and as a structure, it's just a little ball of cells. It's really interesting to consider that these initial cells already demonstrate the beginning stages of differentiation into tissues and the morphology of the mature organism. Now think back to the egg cell. It's a big spherical or oblong shape with cytoplasm filled with enzymes and sugars and nucleic acids and stuff like that. Many of these molecules serve a regulatory purpose, and their position in the egg cell's cytoplasm is no accident. When the egg divides, the cleavages that form and separate new daughter cells from each other are accurately aligned so as to properly arrange those regulatory molecules. For example, say that there's a certain type of regulatory molecule that exists in a loose concentration in the cell's lower left limb, like say just in that particular region of the cell. Well, when the egg starts to divide, and the whole thing breaks up into smaller and smaller daughter cells, all of those daughter cells that are produced from that lower left quadrant, that lower left region of the original cell, they'll all contain some of the original cytoplasm from that area, along with all of those theoretical regulatory molecules that were just hanging out in a loose cloud there. So this is an example of how the chemical arrangement of proteins themselves in the very initial cell of a new individual animal, that plays a fundamental role in the embryonic development of the new animal. Now, when development continues and more genes start getting expressed, these regulatory molecules will alter that gene expression in some way, and tissue differentiation will begin in earnest. These regulatory molecules will control the transcription cascades and protein expression of their cells, which will alter how they grow, what they differentiate into, what they do, what chemicals they express. It really does lay down the groundwork for complete tissue differentiation, which, as you'll see, is extremely important to the animal organism. Now, after a few days of rapid cellular division, the blastula has now become a mass of cells, all primed with a particular set of genetic regulators. This mass of cells typically has a cavity, or a fluid-filled space, inside of itself called a blastocele. So the actual cells take the shape of a racquetball or something like that. Because it's spherical, but if you were to cut it in half, you would see that there's a big cavity inside, a big open space. And this is called the blastocele. Now when this forms, when the blastula has taken this shape, where it has a defined blastocele, at this point the blastula will enter a phase called gastrulation, where the cells will reorganize themselves in a careful dance and settle down in 3 distinct layers, called germ layers. This cellular organization fundamentally establishes the body axes of the animal, where its head will grow, where its limbs will grow, how it will be oriented, where its symmetry will be, and animals tend to be bilaterally symmetrical. There's a lot of animals that are radially symmetrical, like jellyfish and starfish, but a lot more are bilaterally symmetrical. These newly created tissue layers, or germ layers, are the ectoderm, the mesoderm, and the endoderm. Now the prefixes kind of give it away, but the ectoderm is the outer layer of cells, which wraps almost entirely around the gastrulating blastula. The mesoderm forms the second layer immediately beneath the ectoderm, and the endoderm tissue fills in the inner space. It's the inner lining. The the growing endoderm will fill in the blastocele, but it will form a new internal cavity somewhere else, which will become the animal's gut, or its stomach and intestines. This cavity will become the inside of the animal's food tube. This endoderm tissue will go on during development to form the inner lining of the body's tubes and organs, like the urinary tract, the digestive tract, and the respiratory tract. The endoderm also produces differentiated cells that grow into many important internal organs, like the liver, the lungs, and the thyroid, among many others. Now, the mesoderm, which is the middle tissue, grows into all of the internal stuff that's between our digestive tract and our skin. The mesoderm produces differentiated cells that grow into bones and muscles, into veins and arteries and lymphatic structures, and all the outer covering of most of the organs. The mesoderm also forms the genitals and the dermis layer of the skin. The third layer, the ectoderm, produces cells that are widely differentiated. As you would expect, the epidermis of the skin is produced from the ectoderm, as is the tissue lining the inside of the mouth and the rectal cavity. The cornea and the lens of the eye are also produced from ectoderm. Now, this might be surprising, but part of the ectoderm actually sinks deep within the developing organism and produces the brain and all of the nervous tissue that innervates the body. After gastrulation, these mesoderm layers have been established, but none of the organs have actually been produced yet. This occurs through a process called organogenesis. The cells in the germ layers of the blastula will divide and reorganize again, and they'll undergo more differentiation to become very specialized tissues. These specialized tissues form nodes that grow in size, and they become the various organs and structures of the body. Consider chordate animals as an example. The chordate animals have a notochord, which is a rod-shaped mass of mesodermal tissue that forms along the back edge of the embryo. The notochord is like a scaffolding that helps to organize other embryonic structures and coordinate development. Some invertebrate animals retain their notochords throughout their life, but vertebrate animals, their embryonic notochord will eventually dissolve. It'll dissolve in the embryo. 3 structures are formed that run parallel alongside the notochord. There's some more mesoderm tissue that will condense to form 2 long columns of cells called somites that run along either side of the notochord, along the animal's back. As these somites form, the ectoderm layers above them will form parallel crests, or ridges. These will continue growing upwards until they curl inward to meet each other and form a tube. This ectoderm structure is the neural tube, and it will go on to become the animal's spine and its brain. So you can see how this process allows the outer ectoderm tissue to fuse into a tube, which can sink inwards deeper into the mesoderm, where it will then become the animal's nervous system. Much like the vertebrate notochord, the somites are also temporary. They'll break up into differentiated chunks of tissue that will become muscles and bones and the outer lining of the organs and the dermis of the skin. Early in this stage of development, cells can be moved around to different areas, and they'll quickly adapt and blend into the specific differentiation of the cells around them. This is to say that transplanted cells acquire the specialization of nearby cells. They're like stem cells. However, this plasticity will fade as development continues, and differentiating tissue begins to settle into their place, and it becomes determined. A differentiating cell reaching determination can no longer change its differentiation or its specialization. This is to say that it can't change the patterns of gene expression that will give it its unique functional and physiological qualities. Remember that this differentiation begins all the way back in the beginning, with various regulatory molecules floating in particular regions of the cytoplasm in the original egg. At this point in development, the cellular precursors for all of the body's tissues have been positioned correctly, and their genetic expression has been specialized and sufficiently differentiated so that they have started making organs and bones and the brain and nerves and everything else. Everything is in place. All tissues are growing and swelling in size and perpetually increasing in complexity as the cells divide and further differentiate into every kind of tissue. In many species of animal, these embryos are grown inside of eggs, which are deposited by the female into a nest, or some kind of protective chamber or safe area where they can grow and eventually hatch. These animals that lay eggs are oviparous. They have internal fertilization, which produces embryos in the eggs, and the eggs are then expelled. Birds, for example, are oviparous, and a big part of their life involves building nests to hold, watch over, and protect their eggs. Reptiles are also oviparous, but they tend to be a little bit more detached. In many species, not all of them, but in many species, they'll bury their eggs in sand or dirt, and then leave, letting the young hatchlings fend for themselves. The embryos in these eggs survive and grow by feeding off of the nutrients contained within the egg's yolk tissue. Other animal species are ovoviviparous, which means that the organism produces eggs like an oviparous animal, but instead of depositing them outside of the body, the parent will retain the egg on their body. However, the egg feeds exclusively from an embryo, and it doesn't take any nutrients from the mother. There's other animal species that are ovuliparous, which means that eggs are produced unfertilized, and they get released into the surrounding environment where a male fertilizes them externally. This is typically the case for aquatic animals, like bony fish, cnidarians, and echinoderms, like starfish. Now, some organisms don't grow in egg structures at all. The young are grown free-floating in the female's oviduct. These animals are called viviparous, and there's two types of viviparous reproduction. Hemotrophic viviparous animals produce their young in the uterus, and they feed them with nutrients coming directly out of the mother's bloodstream. What the mother eats and digests, the fetal offspring growing in the uterus will also ingest. This nutrient exchange occurs through an organ like a placenta. Humans, and most other mammals for that matter, are hemotrophic viviparous animals. This is how we do it. This is how we've evolved. Now, the other kind of viviparous animals are the histotrophic viviparous kind. And these are grown in the uterus, but they don't feed off of the mother's blood nutrients. Instead, they literally cannibalize their embryonic siblings in the womb, eating up all of the other nearby zygotes and embryos and anything that is basically a floating mass of tasty nutrients. This brutal form of fetal development exists in a small range of species, and most of them are sharks. In viviparous animals, like humans and most mammals, when the young are being grown inside the uterus, they're going through a period called gestation. I'll talk more about gestation and the nitty-gritty biological details of gestation in humans in a future episode, but for now, just understand that gestation is the period in development where the new organism develops from an embryo and forms a viable fetus within the mother. Gestation ends when the female gives birth. The offspring is born into the world, where it opens its eyes for the first time, and takes its first breath in the big, wide world outside the womb. Okay, so I want to change gears here. I've just gone through a brief overview of animal development as a process, but now I'm going to explore what is being developed and why. This means the animal's physiology, its limbs, its mouth, its eyes and ears, its body plan, and the associated behaviors and life strategies that the animal uses because of all of these morphological variables. To begin, I'll have to go back in time, to the very beginning of the animal lineage, before all of this variation and diversity in the animal kingdom had ever been created. Now, the first evolutionary milestone that began to shape the emergent animal lineage was the emergence of multicellularity, more than 600 million years ago. The first multicellular organisms were masses of tissue that absorbed food directly out of the ambient water, and so they became the first sponges. The sponges, or the porifera, are the most primitive, simple animals. Some species have an extremely thin epithelial layer, which lines the inner and outer surfaces of their bodies, and their bodies themselves are evolutionarily designed to be just a mass of pores and channels. Water will flow through these pores, through these channels, and food particles are extracted, they're filtered out and ingested. Unlike most other animals, sponges only have one germ layer, not three, And while there is some cellular differentiation, sponges don't really form true tissues or organs. Despite their relative primitiveness, the sponges demonstrate a few things about the animal lineage, showing off the qualities that were there at the very beginning. These qualities, these traits, include a primitive level of cellular specialization, programmed apoptosis as part of development, the use of flexible sheets of connected cells, tissues, and a capacity to recognize cells composing the self as opposed to cells from the non-self, or cells from other organisms in the environment. From within the lineage of sponges emerged the first diploblasts. These were the first animals with 2 germ layers in their embryonic stage, and these germ layers possess cells that undergo significant differentiation to form true tissues. These diploblasts have an endoderm and an ectoderm, and they form the inner and outer lining of the organism's body, respectively. The diploblasts tend to be simpler animals, like ctenophore comb jellies, and cnidarian animals like sea anemones and jellyfish. These animals don't have a centralized nervous system, and they don't really have sophisticated bones and muscles. Instead, they have a decentralized network of nervous cells, and some species have inflated cells, or structures that fill with water and act kind of like a hydrostatic skeleton, or kind of like a water balloon. And they have thin muscle cell analogs that help the organism twitch and spin and numbly move their tentacles around. They don't really have a whole lot of internal organs either. Between their inner and outer layer, they're filled with a simple mass of jelly-like goo called mesoglea that gives them a bit of volume and a little bit of structural integrity, even though it's really fragile. They're really like water balloons filled with jelly. From the lineages of diploblasts emerged an ancestral organism with a 3rd germ layer, which was the mesoderm. Now this created a lineage of triploblasts because of the 3 germ layers, and this opened the door for the evolution of much more complex organisms. This mesoderm led to the evolution of complex and powerful muscle tissue, as well as the architecture of a sophisticated vasculature and powerful organs. Also worth noting is the shape and the symmetry of the animals. The early animals, like jellyfish and sea anemones, are like domes and cylinders, shapes that are circular. These organisms are radially symmetrical. They look the same no matter which side you're looking at, the front or the back or the left or the right. They have a top and a bottom that you can differentiate, like the top of a jellyfish versus the tips of its tentacles, but they don't really have a front or a back side. Well, triploblast organisms evolved a bilateral symmetry. They have longer, narrower bodies, with a definite right side and left side, and a specialized front side and back side. This major shift in body plan allowed animals to move far faster than they could previously, and this sped up the development of complex food webs and ecologies, and with that came the evolution of complex mouth parts, feeding parts, and all manner of sensory organs to detect and track prey and to avoid predators. Also, the decentralized nervous systems of the diploblasts coalesced into a centralized nervous system in the triploblasts. The diploblasts can use a decentralized neural net rather effectively. Because of their radial symmetry, food can approach them from any angle, so having a diffuse neural net allows for maximal, localized responses in the food-capturing limbs, like the sea anemone's tentacles. However, in the triploblasts, with their bilateral symmetry and mouth orifice at a distinct front end, a centralized nervous system gave them a sensitive head region for optimally detecting and finding food. The body trails behind the head and mouth region, with the centralized nervous system rooting through it in the form of a spine, which branches off a peripheral nervous system that innervates the flesh and the limbs and the organs and provides sensation and automaticity to the organism. Now, inherent in this evolutionary process is the cephalization, or the development of a head region in the first place. This head region is at the front of the organism, so it's the first part that goes wherever the animal is going. It's the first part to run into food, and to see out in front of the organism unobstructed. It's for this reason that an animal's head became a particularly important structure, packed with all manner of sensory equipment and orifices for eating and respiration. The body is positioned behind the head, and the limbs coming out of the body allow for the organism to carry itself or propel itself through the environment. It can search for food, or chase after food, or run from predators. Consider the fact that these early triploblastic animals were all aquatic and soft-bodied. Within them, the mesoderm evolved an internal fluid-filled cavity called a coelom. Now this created a hydrostatic skeleton that gave the animals stiffness and rigidity, and this in turn sharply improved their motility in the water. The coelom also provides an internal space for nutrients to float around, as well as for the gut to move and flex within the body, as the animal's outer meso and ectoderm layers bend and twist as it, engages the world around it. If the gut was stiffly attached to the ectoderm with a solid mass of mesoderm, that would limit the flexibility and the mobility of the animal. The coelom loosened them up and provided the flexibility for fast, twisting, wiggling body motions. Okay, so where are we now? I've talked about a lineage of animals that has evolved a centralized nervous system, a head structure, and a coelom. These are the bilaterian animals, so named because they all display bilateral symmetry, which is perhaps the foundational quality from which all other qualities are derived. Within this bilaterian lineage, two groups emerged, called the Protostomes and the deuterostomes. In the near future, I'll be releasing episodes that will explore both of these groups in vivid detail, but for now, I'll just stick to the basics. The titular difference between the protostomes and deuterostomes takes place during gastrulation. In the protostomes, the hole in the germ layer that becomes the mouth will form first, and then the anus will form second. Furthermore, the coelom itself is formed when chunks of mesoderm cells grow into a large mass, and then this mass hollows itself out from the inside, either through apoptosis or from the cells migrating and moving out of the way to create this internal cavity. This will leave behind a void that gets filled with fluid, and it will become the coelom. In contrast, in deuterostome blastula development, the hole in the germ layers that becomes the anus is the first to form, and the mouth orifice forms second. The mesoderm doesn't form blocks of biomass that hollow out to form the coelom, but instead, the mesoderm will bubble out from the gut, and it pinches off to seal in a cavity, which fills with fluid to become the coelom. These two different developmental patterns eventually form the same end structure, which is a body with a coelom, but they represent some of the earliest and most basal differences between some of the largest divisions in the animal kingdom. This is the evolutionary condition of the animals 400 to 500 million years ago. When you start looking at the animal kingdom at the level of protostome and deuterostome, you start to see a wildly diverse and dynamic range of organisms, all of them evolving new traits and losing old ones. For example, the platyhelminths, or the flatworms, evolved to lose their coelom. The echinoderms, like starfish and related species, lost their bilateral symmetry and re-evolved a radial symmetry. There's other trends that were particularly advantageous, which led to them being evolved independently across multiple lineages, like the eye. There's all sorts of different eyes across the animal kingdom because an eye structure was evolved independently multiple times. It's a very curious case of convergent evolution that there's distinct types of eyes based on their structure. Like lensed eyes, for example, are a particular type of eye that's very effective at forming images, and the evolution of a lensed eye in whatever given species or group of species has happened independently multiple times throughout the history of life. One of the more obvious and basal traits of this nature is segmentation, which was evolved independently in annelids, arthropods, and chordates, and in some lineages of mollusks. Segmentation is a really cool process, and it's really trippy to think about, because it brings me right back to the food tube metaphor. Animals are bodies built around a tube, which is meant for digesting food. A genetically efficient form of body planning was evolved and preserved, wherein a small set of genes could be expressed at different points along the tube to create repeating body structures. At each point of limb or appendage formation, the gene expression could be similar to that at nearby points, or it could be slightly different. This gene expression, and the differences from point to point, from segment to segment along the tube, produces limbs with certain qualities. A small toolbox of genes can thus be regulated, its expression can be regulated in different ways so that it expresses certain genes in a certain amount, and the end result is a controlled development of different types of limbs, like your arms, hands, and fingers, and your legs and feet and toes. All of these limbs are generated with a very similar set of genes, but because of the slightly different expression of those genes, this leads to your legs being larger and stronger than your arms, and it leads to your arms being more flexible and tactile than your legs. And it also controls the development of your hands and fingers, and why they're so much more dexterous and sensitive than your toes and feet. In contrast, consider an insect like an ant, which has 6 very similar legs. The gene expression to produce each leg is very similar to that used to produce another leg. But then again, these legs are entirely different from the mandibles and antennae produced on the head, even though the production of these structures involves many of the same genes, but a radically different gene expression. The head, thorax, and abdomen of an insect are all body segments where specialization of gene expression has been streamlined. This is very much the same way that your head, torso, and pelvis are distinct body segments, with assorted limbs and appendages protruding from each one. The number, the size, shape, and the function of the appendages are all based on the segmentation, on these slight alterations to your gene expression along the length of the animal's body. This in itself is a result of natural selection searching for an optimal biochemical efficiency. This method of body generation offers extremely high morphological plasticity relative to the number of genes involved. You can have just a small number of genes, but if you just express them differently, you can create this huge variety of different structures with all these different uses. Evolutionarily speaking, it is incredibly efficient, and it's no wonder that this was evolved multiple times, and in virtually every lineage where it was evolved, that trait has persisted. Segmentation is really important in that sense. In addition to offering high morphological plasticity, it also suggests that morphological changes over evolutionary lengths of time may not necessarily be due to mutations in the genome itself, but instead could be due to changes in the pattern of gene expression, like changes in the animal's epigenetics. All of these limbs and appendages and various structures growing off or inside the organism, they all serve some purpose that helps the animal survive and propagate its genes through time. Sensory organs detect the ambient environment, and they help an animal stay aware of its surroundings. Light-sensitive cell structures can form eyes, and these help the animal detect light wavelengths and see its environment. In the most primitive sense, you have microscopic animals and various mollusks whose eyes are hardly more sophisticated than little clusters of light-sensitive cells that can tell the difference between sunlight and a shadow. And then on the other end of the spectrum, you have very sophisticated lensed eyes that can produce highly detailed, colored images of the world around it. You also have sound-sensitive cell structures that form ears, and these help the animal detect various waves in the air or the water that are perceived as consistent and recognizable sounds. Chemically sensitive cell structures can form tongues and noses and all manner of strange equivalent chemosensory organs. Some animals have evolved really crazy sensory structures that give them enormous fitness advantages. For example, some animals have magnetic sensitive organs that can detect magnetic fields, which is useful for navigation during long migrations, like if you're a turtle just wandering around in the ocean. Some animals, mostly predatory animals, have electrically sensitive organs that are capable of detecting the bioelectrical activity in the muscles of nearby animals. Try being a little fish and hiding from something that can smell the electric activity in your cells. Good luck hiding from that, you're probably not going to succeed. Now, these are just some of the most common sensory organs and structures in animals. There are many more with a wide, diverse range of sensory abilities. Alright, so I've been describing animals when boiled down to their most fundamental qualities, as mobile food tubes. There's two important things that are implied here. The first is that the animal can move, and the second is that the animal eats stuff. So, how does it move? How does it eat stuff? Like everything else in the animal kingdom, the physiology is diverse. Limbs meant for movement are believed to be homologous in the kingdom, which is to say that the common ancestor of all limbed animals had a rudimentary set of limbs, and alterations to those ancestral genes and gene expression has provided all of the variety in limbs in the animals that we see today. There's lobed limbs, which are little stumps used to grip and crawl. There's tube limbs, which are like numerous long, thin tubes that use suction and hydraulic pressure to pull the animal across the surface. There are slippery, twisting tentacles and arm-like limbs, like those you would find in squid and octopus. And there are jointed limbs, like you see in arthropods and chordates. Some of these jointed limb animals have evolved wings, and they can fly through the air. As for feeding, the appendages and morphologies, and even the general feeding strategies, are all just as diverse. Corals and sponges are filter feeders, which means they extract food by filtering it out of a liquid matrix. Baleen whales are also filter feeders, which extract krill out of vast gulps of ocean water. Animals like sea cucumbers are deposit feeders. They will creep and crawl along the forest floor, or the ocean floor, where they feed off of layers of deposited detritus and waste. And then there are fluid-feeding organisms, like moths and hummingbirds, which have tubular mouthparts used to suck up nectar from plants. Most animals, at least I would imagine most animals, are mass feeders. Humans are mass feeders. Lions are mass feeders. Cows are mass feeders, as are pretty much any other vertebrate and most arthropods that you can think of. Mass feeders have evolved sharp mouthparts, like fangs or mandibles, which tear chunks of biomass off of their prey and bring it into the animal's digestive tract. Lastly, animals produce appendages and internal body structures that are used for reproduction, for the perpetuation of their species. Corals and such are asexual, and they can reproduce by budding, but most animals are sexual, and they need a mate with complementary genitals and complementary gametes in order to reproduce. The male typically has external genitalia, which it uses to penetrate the female and deposit sperm into her internal reproductive structures, which will fertilize her egg and produce a new individual animal. And with that, we've come right back to the beginning. We've come full circle, starting right again with the blastula and embryonic development. Alright, so that's about it for this episode, everyone. If you enjoyed it, then stay tuned for the next episode, where I'll explore the evolutionary relationship that animals have with water. We'll be diving headfirst into the biology of animal hydration, so be sure to check it out. And as always, thanks for listening.